formed elements of the blood consist of red blood cells, white blood cells, and platelets. We're going to focus on the red blood cells. The anatomy of a red blood cell is really quite interesting in that it is described as a biconcave disc. What this means is that both sides of the red blood cell are sunken in. These red blood cells are only about 7 micrometers in size. And when you think about the size of a micrometer, there's 1,000 micrometers in a millimeter. And this is still just a little bit larger, actually nearly twice the size of the smallest capillaries. Now, what does the biconcave disc do for a red blood cell? Well, one thing is that it provides a very large surface area so that oxygen and carbon dioxide can very easily and rapidly move through the cell membrane to bond with hemoglobin within. It enables the red blood cells to stack in a capillary or maybe just a slightly larger vessel. You see right here the stacking nature. What that does is keeps the cells from bouncing around in that blood vessel, and then all of them move through the vessel at one time. Thirdly, it enables flexibility of the red blood cell. It allows it to twist and turn when it gets into those capillaries that are about 1/2 of their size. Red blood cells are anucleate, meaning they do not have a nucleus. The implication of this is that, number one, they cannot reproduce. Number two, it allows for them to carry more hemoglobin, like hundreds of millions of molecules of hemoglobin. And lastly, it has a very limited lifespan of about 120 days. Yeah, about 4 months. Focusing on the hemoglobin, this is a protein molecule. It is a quaternary structure, meaning that it has several protein chains bonded together. 2 of these chains are alpha chains, and the other 2 are beta chains. At the center of these chains is a heme unit. The heme unit consists of an iron atom in the center. It is to this iron atom that oxygen molecules will bond. Because there is a heme unit for each of the chains of hemoglobin, we can bond 4 oxygen molecules. 1, 2, 3, and 4. Now, when those 4 hemes are bonded to oxygen, we call this molecule oxyhemoglobin. When it is empty, it is called deoxyhemoglobin. Now, carbon dioxide can bond to the heme unit as well, and when carbon dioxide is attached, we call it carbaminohemoglobin. Just a side thought, there are like about 280 million hemoglobin units. Okay, so we have a short lifespan. How do we replace our red blood cells on a regular basis? Erythropoiesis is the process that is used. Now, in erythropoiesis, we begin with hemocytoblasts that have been stimulated to myeloid stem cells, and then when erythropoietin, EPO, is present, they can differentiate into the mature red blood cell. So about day 1, we have a proerythroblast. At this point, our cell is simply growing. Notice here too, we have a very large nucleus present. From here, we move into the erythroblast. This is day 2 to day 4. And what's happening in our cell now is a lot of hemoglobin production. Remember that protein synthesis relies on having a nucleus with DNA present in order to get the mRNA, which will be translated into a protein. About day 4, our normoblast ejects its nucleus and becomes a reticulocyte, existing about days 5 to 7, still in the red bone marrow. At this time, the hemoglobin is still being synthesized. mRNA is still present in the cytoplasm, and so our cell can continue its hemoglobin synthesis. At that point, the reticulocyte is released to the bloodstream, and from there, they will mature into the red blood cell. Lifespan is only 120 days. What has happened? This cell has been through countless miles of blood vessels. Consequently, they're a little bit on the ragged side, and they don't have the means to repair themselves. At this point, in the spleen, liver, and red bone marrow, macrophages are going to phagocytize the red blood cell and break apart the globins or globulins, globular proteins, and break them into amino acids that then are going to be recycled, perhaps for use in red blood cells again, perhaps not. Perhaps it will become a different protein. The heme unit is also separated off, and the iron is separated from that, and it is going to be recycled in the red blood cells. Now the remaining parts of the heme unit undergo chemical changes. First, it's a green pigment called biliverdin, and from there it becomes bilirubin. Bilirubin is usually pretty familiar to all of us because of jaundice, usually associated with liver diseases of some kind, as well as newborns who have a little difficulty digesting certain proteins. Bilirubin is a yellow pigment. Now, bilirubin is going to be transported to the liver, and it is excreted in bile. Then it undergoes chemical processes in the digestive tract into urobilins, which then will be absorbed and taken to the kidneys and eliminated there in the urine, or stercobilins, which will be eliminated in the feces. Both of those have a brown color.